Photosynthesis is a natural process that plants use to create sugar. It all happens in the chloroplast, specifically the thylakoid membranes, located in the thylakoids. We can see that the membrane has photosystems 1 and 2, as well as channeling proteins, electron transport chain, protopump, and ATP synthase. Outside the membrane is the stroma, and inside is the thylakoid space. The reactions begin when light hits photosystem 2 and charges the electrons, making them excited. The excited electrons move forward to the electron transport chain onto the channeling protein. Water gets split into 1 oxygen, 2 hydrogen ions, and 2 electrons that travel up to photosystem 2, replacing the missing electron. As excited electrons move down the proton pump to another channeling protein, their energy is used to pump protons from the stroma to the thylakoid space. The electrons are no longer excited because they have lost their energy. This process creates a high concentration of hydrogen ions inside the thylakoid, so the ATP synthase allows the extra protons to passively diffuse from high to low concentration. Protons cause ATP synthase to spin as they move up to the stroma, creating ATP. ATP is one of the most important products of light-dependent reactions. As soon as electrons reach the end of the electron transport chain, they move up to photosystem 1, where light excites them again. They travel down another transport chain, where molecule NADP+ meets them at the NADP+ reductase. When NADP+ accepts electrons, it also accepts hydrogen and becomes NADPH. The NADPH carrier is also one of the most important products of light-dependent reactions. It carries electrons and hydrogen ions to the Calvin cycle. Carbon fixation. Carbon dioxide from the atmosphere is captured and incorporated into 5-carbon sugar molecule, or UBP, with the help of the enzyme Rubisco. The initial incorporation of carbon from CO2 forms 2 molecules of 3-phosphoglycerates, which are 3-carbon compounds. Reduction. ATP and NADPH, generated during the light reactions of photosynthesis, are used to convert 3-PGAA into a more energy-rich 3-carbon sugar called glyceraldehyde 3-phosphate. For every 3 turns of the Calvin cycle, 1 molecule of G3P is produced, and the other 5 molecules of G3P are used to regenerate our UBP. Regeneration of our UBP. The remaining 5 molecules of G3P undergo a series of reactions that require ATP to regenerate 3 molecules of RUBP. The RUBP molecules are then available to initiate another round of carbon fixation, ensuring the continuous cycle. In summary, the Calvin cycle begins with the fixation of carbon dioxide, proceeds to the reduction of 3-PGA to G3P using energy from ATP and NADPH, and concludes with the regeneration of RUBP. This cycle is crucial for synthesizing organic molecules, especially glucose, which serves as an energy source for the plant and other organisms.